Hello. Good afternoon, everybody. Welcome to the Biodiversity Conference. Please silence your cell phones. Hope you guys have been enjoying yourself, having a good day. Our next speaker has been a UC Berkeley professor of herpetology for 19 years. His research has him knee-deep in African swamps as often as it has him in the lab, which makes for a pretty exciting presentation and career. His work has revealed some important links between conservation and public health and has given him the opportunity to speak out publicly against the EPA and the safe level limits that they've set on many of the toxins that we find in our public drinking water. So with no further ado, Dr. Tyrone Hayes, UC Berkeley.
Thank you. I want to thank you all for coming. Thank you all for having me and allowing me to be a part of what, at least for me so far, has been a pretty exciting event, pretty exciting day. I'm going to talk to you today about a talk. It's a summary of a keynote that I gave at the World Congress of Herpetology just up here in Vancouver earlier. I noticed on the program it just says my title is Amphibians, which probably means that I didn't answer an email that I was supposed to, and I apologize for that. But here's my title for today, The Cause, and it's meant to be an obnoxious and sarcastic title, The Cause of Amphibian Decline, as if we've only done one thing to screw up the planet. Before I begin, though, as always, I want to acknowledge and thank the people who are most important in my life and the people who've made this possible. As usual, my first acknowledgement and thank you, I do my thank yous first. That way when I run out of time or the chemical manufacturers come to get me, I've done the important stuff. But as usual, my wife in the middle, Catherine Kim, and my son Tyler and my daughter Cassina for their love and support. My family at large for their love and support, continuous love and support. I want to thank my funding sources, because science doesn't get done without money. And also, this is a matter of disclosure. I have been funded by the chemical industry, Novartis, Syngenta, Crop Protection, and EcoRisk, but they no longer like hanging out with me. But I'll give you some of their perspectives throughout my talk, so-called, I guess, words from my former sponsor. Then I want to thank my students. The students that have been involved in the work are many, and everybody in blue is an undergraduate, and that's been a very important aspect of my laboratory, my career. I had a wonderful experience as an undergraduate doing science, and I've been trying to give that back as best I can. These are my current lab members, and they've done some of the new work that hasn't been published yet, and I'll talk about some of the work that they've been doing as well. And finally, as always, since 2005, this and all of my talks are dedicated to my grandmother, who, luckily before she passed away, she passed on her love of teaching and her desire to make the world a better place through science to me. She's also the person who taught me that you don't lecture and you don't give talks. If you really want people to listen, you just tell a good story. And so I'm just going to tell you a story today. My story starts with a little boy who likes frogs, and this slide is a picture of a book that my mom sent me when my son was born. And she wrote a note in the book, and it said, "This was your dad's favorite book." And as I always tell people, I don't remember the book at all, but my mama don't lie. She said it was my favorite book. It was my favorite book. What I remember, though, is that I've been after this question for as long as I can remember. What is a frog? For as long as I can remember. And from that time, I'm really just a little boy who likes frogs. So that's what this story is about. It's about literally just a little boy who likes frogs. Now, one of the things that this little boy dreamed about, also for as long as I can remember, was going to Africa. I remember, my family grew up, our family of five, we made $9,000 a year. The first time I ever got on an airplane was when I went off to college. So it was truly a dream that I never thought would see come true. I remember folding out the National Geographic magazines and dreaming about going to this magical place to look for frogs and see the animals. And that dream came true, actually, through National Geographic. So literally, I got to become the guy in the magazine and on the TV show, and I even got to do a commercial with a Toyota truck driving on the thing. It was not only and we're going in chronological order, because the first time I got to go, we started with the little boy who likes frogs. The first time I got to go to Africa, I was a graduate student. And I went there to study frogs, funded by National Geographic. And while I was there, I discovered a little frog that would drastically change, even though I didn't know it at the time, the trajectory of my professional career and really my life. This is Hyperoleus argus. Sorry, it has no common name. It only has a Latin name. That's how little known it was. And what's fascinating about this frog, especially to a little boy who likes frogs, is it's pretty. The males and the females are differently colored. They almost look like they're different species. So I brought these animals back to the laboratory and was trying to discover why the males and females look different. And we found that when we raised them in the laboratory, so this is the same individual, photographed once a day for 6 days, that when we raised them in the laboratory, they all start out green, and then the females change color at puberty or at sexual maturity. So I sort of surmised then that this might be hormonally regulated because it wasn't genetic, otherwise they'd all do the same thing. And we found that testosterone, the so-called male hormone, didn't make them change colors. But if we literally just dipped the little frogs in estrogen, it would cause their color patterns to change. So the female hormone estrogen actually had a color-changing effect on these animals. And so here's the species Hyperoles argus. What happened after that is really the thing that really changed my life. It was February 15th in 1993, and I remember exactly the day because it's the day before my son was born. I was giving this talk up at UC Davis on this color-changing frog, Hyperoles argus, and my wife started going into labor. It was the day before my son was born, you see. And we were driving back down the highway, highway coming down 5 there, and my wife, between contractions, said, she has an MBA and an MPH. She said, you should patent that frog. And I said, what do you mean I should patent that frog? That's crazy labor talk, And she called her brother, and her brother said, no, you can patent a frog. Heretofore to be referred. So he's a lawyer, right? He was talking that kind of stuff. And so we did. We called it the Hyperoles argus endocrine screen, or the Hayes test. Changed my life. Here's why you patent a frog, first of all. Here's a control, untreated animal. They all start out green. If you give them estradiol, which is a natural estrogen that's in all animals, doesn't matter if you're a frog, a dog, cat, or a hog, if you're a female, you have estradiol in your bodies. If you give them methinyl estradiol, the estrogen that's used in birth control pill, they'll change color. If you give them DES, a very potent estrogen that's used as a pharmaceutical, they'll change color. If you give them DDT, which is not a hormone, it's a pesticide, but it actually binds the estrogen receptor, they'll change color. And that's why you patent a frog. Because see, every compound we tested that was estrogenic in our frog was also known to promote breast cancer in humans. So we had a little frog, and we tested dozens of compounds. We had a little frog that we could raise by the tens of thousands, and we could dip them in different solutions and determine if those compounds might be breast cancer promoting agents, or you could mail me a sample of your water, and I could dip the frog in it. If the frog changed color, I'd say, hey, you might not want to drink that water. What's more is we found out that if we treated the frogs with tamoxifen first, the estrogen didn't work. Tamoxifen blocks the estrogen receptor and is used as a treatment for breast cancer. So we not only had a frog that could help us find out what compounds might promote breast cancer, we had a little frog that might help us determine what compounds might help block breast cancer. Then things changed. Then this little boy who likes frogs finds out about something called intellectual property. You see, anything that I think of, as long as I'm being paid by the university, belongs to the university. So the university says, little boy who likes frogs, after you sign on the dotted line, that property belongs to us. And they said, unless you find a market, unless you find a place that you're going to sell your idea, then we're going to take it and give it to somebody else. I'm just a little boy who likes frogs. But that's what introduced me to the scarlet letter, atrazine. See, atrazine is this compound that's manufactured by the largest chemical company in the world. At the time, it was the #1 selling pesticide in the world and the #1 contaminant of drinking water in the world. Here's what it looks like. It's an S-chlorotriazine. It's an herbicide or weed killer that's primarily used on corn, but up here in these parts, it's mostly used in forestry. It's been used since 1958, and we use 80 million pounds in the United States per year of atrazine alone. It's used in more than 80 countries, but interestingly, it's outlawed in all of Europe. And now I put this incorrect, misleading slide up here on purpose. At least that's what the company lawyers say. See, according to them, it has not been outlawed in all of Europe. It has been denied regulatory approval by the European Union. So whatever difference that makes, as long as it keeps upsetting the lawyers, I'll keep putting this slide up. The main point being that the company is based in Switzerland. So we're using 80 million pounds of a chemical that's not allowed on the continent that the company calls home. So what we ended up doing was looking at another African frog, and actually this is the South African clawed frog, and in fact we collect them in San Diego, so I guess technically they're African-American clawed frogs, but at any rate, it's an interesting little frog because this is the frog that was originally used as the pregnancy test. So believe it or not, in 1920, if you thought you were pregnant, you would go to the doctor and they would inject some urine into the frog, and the frog laid eggs. Well, then you were either happy or sad, depending on your situation. Here's why I like to tell this story. One, isn't it interesting to imagine, just imagine, what was going through the guy's head, the first guy who thought, I wonder what will happen if I inject pee into a frog. Where does that idea even come from? Where does that come from? The second reason I tell the story is the following. In the same way that the same estrogens that cause my frog to change color tells us that our hormones are so similar to frogs that if frogs are affected, we should be worried. In the same way, the hormone that maintained the pregnancy that's responsible for everybody in this room will also make this frog lay eggs. Our hormones are so close that if I often call this a tale of toads and men, playing on Steinbeck a little bit. As I tell this tale of toads and men, you should be wondering, if this stuff is messing with the frogs, what is it doing to me and my children? And that's where we'll end up today. So the first thing we showed in our experiments was that atrazine inhibited growth of the voice box or the larynx in males. So the males couldn't sing properly. Once we discovered that, we figured, well, the next place to look are the gonads. Because you see, male frogs sing and females don't because of testosterone. The same reason that men have deeper voices, a larger voice box than women. So it's bad news for the company. And they were paying for the research at the time. The gonads of frogs look like this. So here are the kidneys, okay? Those are testes. So this is a male. Oh, but wait, there's some ovaries. Oh, wait, there's another testis again. Oh, wait, there's more ovaries. Like a whole party going on in there. You never even have to leave your own body. It's not normal. And that's not a judgment call. All I'm saying is I've been giving this talk for like 10 years, and every time I give it, somebody in the audience says, aren't frogs naturally hermaphroditic? And I finally asked, why do people think that? Who knows? Jurassic Park, yeah. So apparently, I even saw the movie and read the book and didn't remember this. So apparently in Jurassic Park, it was the frogs that were, that's science fiction. Frogs, males are males and females are females. They're not mixed. So here's what we proposed at the time. If you imagine that this big yellow ellipse is your testicle, or imagine a friend, or imagine somebody you don't like, testicles. You shouldn't make testosterone. The word testosterone literally means testicular hormone. Two words pushed together. It's the male hormone, okay? Which males should do. So we proposed if you're exposed to atrazine, this chemical, it turns on aromatase. I want you to remember the word aromatase in case you don't know it. It's the enzyme or the machinery, if you will, that converts testosterone into estrogen. Now, estrogen is the female hormone. The word literally means generator of estrus, which is fine if you're a female, maybe not so much if you're a male. We propose that you're demasculinized or chemically castrated. I like to use that term because the lawyers ask me not to. And you're subsequently feminized because of an inappropriate expression of this machinery to make testosterone into estrogen. Well, all we had to do now is show some evidence. Turns out here's control males, so normal males. Here's atrazine-exposed males, and here's normal females. So their testosterone is no different from testosterone levels in females. So at this time, we're going along, my story's going chronological. So this time, little boy who likes frogs is now coming up for tenure. Y'all know what tenure is, right? That's why I get a permanent job. And we got this paper published in PNAS, Proceedings of the National Academy of Sciences. How many people know PNAS? It's a big deal. So I'm like, I'm going to get tenure now. This is it. I'm publishing. And not only that, another thing that was important, if you look at that, there are 4 black men and a Latino on this paper. That's probably a record for PNAS. So I called my mom up. I call my mom every Sunday. I called my mom up, and I said, Mom, we have a paper coming out in PNAS. My mom drops the phone. 100% true story. She picks back up, and she says, son, how do you get a paper cut on your penis? I said, no, P-N-A. She said, you ain't got to spell it. I used to change your diaper, boy. The next week, my mom calls me up again. And she says, how important is that paper you're talking about, son? I said, what do you mean, Ma? She said, I went to Barnes & Noble and they never heard of that magazine. The point of my story is this. We in the ivory tower put so much emphasis on things that mean nothing to 99.9%. This is one of the top scientific journals in the world. Some argue number 3, and my mom can't go get it at Barnes & Noble. And I'm going to come back later to now how we need to step down out of the ivory tower and do some things that are more important to the public, especially the kinds of things that I'll end up doing. This is now my most important publication. It's a book written for 5th graders, not even written by me, but my mom can go and buy it at Barnes & Noble's, and then everybody can see the kinds of stuff that I'm doing and that their tax monies help pay for. As important as that paper was, however, to me and my career and all that stuff, it didn't solve all the problems we wanted to solve. So, for example, we didn't know if these hermaphrodites were males with ovaries or females with testes, which might sound like a silly question, except that frogs don't have sex chromosomes, so we didn't know who was being affected. We had a pretty good idea because, for example, if we got, in an experiment, we might get 50% females, 30% males, and 20% hermaphrodites. So statistically, we had a good idea, but we couldn't prove it, so to speak. And then we also didn't know what happens when these animals become adults. Again, sounds like an easy question to answer, but it takes these animals about 4 years to grow up. That means you have to get an undergraduate as a freshman and go, hey, I have a project for you, and maybe by the time you graduate, we might have an answer. In fact, it was worse than that. It took us about 8 years, in part, because we didn't know what we were doing. And here's the bottom line. In fact, I did publish this eventually in PNAS again, and I said, wow, this would make a great cover photo. PNAS, the major journal in the United States, said, we're not quite ready for it yet. They published a picture, but not on the cover. And sort of here's why it's important. By the time the paper came out, we had a genetic marker. We could tell males from females. So females express a gene that males don't have, called DMW. So we could tell that this animal's a male, and you see he's kind of smiling. That other one's helping him out because he hurt his leg, for the little one in the front. And that's his brother. It turns out in this population, about 10% of the males completely turned into females. See, he laid eggs. He's a grandmother now, believe it or not. How often do you hear that? About 10% completely turned into females. So I thought, wow, we're ready now for another PNAS paper, something big. But then I thought, if 10% are completely feminized, that can't be the full answer. What possibly could have happened to the other 90%? And so I told the students, I said, well, we're going to study whether the other 90% can reproduce, even though they're still males, already functional. The problem is, these frogs like to reproduce or breed or copulate in the spring, and apparently so do undergraduates. So there's all this discussion about we can't stay here and do the spring. There's all this pool party going on Miami Beach or somewhere, something like that. So in 2008, I made a promise. I said, look, if you stay here and help with this project, you'll get your spring break pool party as well. You got to be creative when you're dealing with undergraduates. Went something like this. That's exactly how this happened. I said, so let's do this experiment. I said, let's put 4 females in here. This is literally the apparatus we use. Then let's put 4 control males and 4 atrazine-treated males and just see if the atrazine-exposed males can compete. A real simple experiment. So when you do that, and here's how that looks. We put them in the pool at 6 p.m., and it's all indoors. And you come back in the morning at 6 a.m. and you look, see they're stitched so we can tell who was who. We can identify them. And then you just look at, who got the hookup. So when you come back and do that, what we found out is the atrazine-treated males almost always lose. And everybody was a virgin. Nobody had experience. Turns out only 2 atrazine-treated males ever won in one of these pool parties. So clearly they weren't functioning properly. What's more is, we wanted to figure out what the hormonal correlates were. So how were hormones involved? So we compared controls and atrazine-treated males from these pool parties, and we found out, as you might predict, that the control males had, on average, much higher testosterone than the atrazine-treated males. What's more important, though, is if you look at who made the love connection, if you look at the individual levels, the atrazine-treated males never got enough testosterone either to compete or to be attractive to the females. The 2 with the hearts are the only 2 that ever won. So there was a hormonal correlate with this failure in reproduction. Now, what we didn't know is, we didn't know if the males couldn't compete with the other males, like are they just getting beat up, or if the females just simply didn't like them. So then we did another set of experiments that I call the Motel 6 experiments. So in this case, we just got them a room, and we just said, you're all alone now with the female, what you gonna do? Males and here's how we assess that. We just let them spend some time alone, and then we let them lay eggs, and then we just count how many eggs are fertilized. So this is a batch of eggs. They lay about 2,000 eggs, and here's ones that's one that's not fertile, and these others show ones that are developing. So yeah, that's right. Real complicated data analysis. There's an undergraduate sitting in a room going 1, 2, 3 for hours. And here's what you find out. A control males fertilize on average about 85% of a female's eggs. So that's not so bad. Atrazine-treated males, only about 15%. And the problems are twofold. One is they don't even try to behave. They don't even try. They sit there and they watch the females lay eggs. There's no competition. They don't even make a move. They don't even try a line, so to speak. The other problem is, even of those that try, here's how the testes of the atrazine-treated males look. Now let me show you what you're looking at. You can probably visually see the difference. If I blow up a section, these are testicular tubules, so these sections, and that's all filled with mature sperm. All those dark heads are sperm ready to go. If I blow up an atrazine-treated animal, you get a testicular tubule that's full of debris, cellular debris and no sperm. So they don't have enough testosterone to show the behavior or to make sperm, even if they do the behavior. Most of their testicular tubules are empty. So then, we published another paper in PNAS with, I think, 8 undergraduate co-authors, and I haven't shown you all the data. We measured a lot more than that. But then we asked them questions about whether or not there were effects in other species of frogs, because maybe African clawed frogs are just weird, and maybe other species won't show these types of effects. We looked at North American leopard frogs in the laboratory, and these guys, so here's a male, that's a testis, and that's a testis, but you see all this, I call it junk in the trunk. You see all the junk in the trunk there? These are all eggs that are bursting through the surface of this male's testis. So again, not normal. Now at this point, Bobby mentioned the EPA, the Environmental Protection Agency, as I was coming up. Somebody here might represent the EPA. If so, you already know that we don't have the best relationship. I called them up at this point because they were evaluating atrazine. I called the EPA up and I said, now I'm looking at North American frogs, because they said they didn't care about African frogs. Now I'm looking at North American frogs. They got eggs popping out of testes. The EPA said, well, that's an interesting finding, Dr. Hayes, but we're not sure if that's an adverse effect or not. Now, on February 16th, 1993, my wife had this G.I. Joe Kung Fu grip on her hand that almost brought me to tears. She tells me that there is nothing more painful in life than childbirth. Got to take her word for it. But fellas, are you with me when I say that I would guess that a dozen chicken eggs in my testicle would have to be somewhere in the top 5, at least, right? The EPA says, no, it's not in our rule book. A frog can have eggs in its testes, and that's okay. So, the other reason we studied the North American leopard frog, however, was we wanted to know if we go outside of the lab, can we find this effect in the real world, or is it a laboratory artifact? So do effects occur in the real world? Now let me give you an example of what's happening in the real world. We get these effects of atrazine at 0.1 parts per billion. That's like 1/1000th of a grain of salt in a liter of water. Okay? The package of atrazine recommends application at 2.9 to 29 million parts per billion. So a farmer might be applying this stuff at levels that are 290 million times higher than we use in the laboratory. To give you an idea of what's in the real world, agricultural runoff can range from low parts per billion up to 10,000. Here's temporary pools, permanent water like lakes and rivers. Here's precipitation, rainwater, up to more than 10 parts per billion. There's enough atrazine in rainwater to chemically castrate and feminize frogs. 500,000 pounds of atrazine come down into rainwater every year. Here's what it takes to chemically castrate a frog, make it into a female. So here's all the environments that would be at risk based on what we know is out there. And here's what the EPA says is safe in your drinking water. 3 parts per billion, 30 times higher than what we're using in the laboratory. And that doesn't mean if you test your water today, it's going to be 3 parts per billion. That means on average, over a year, it might be 100 parts per billion tonight, but on average, it should only be 3. So here's what animals look like when you collect them from the field. These are testes from North American leopard frogs. We're no longer in the lab now. I'm going to show you what's called a transverse cross-section. And all that means is just imagine I'm slicing a salami and folding out a slice. The slice is a different color because of the stain or dye that we use under the microscope. And I'll blow that up, and I'll blow this up. And what you're looking at are testicular tubules that have eggs, that have eggs instead of sperm. This is not in the laboratory now. We're now actually in the real world. And if you go into the real world, here's where most of the atrazine's used, mostly used on corn. Most of this use that's happening up here is in forestry. And we did a transect across the U.S. where we controlled for latitude. And actually, I'm just lying. This is Highway I-80, and we were driving to Indiana to go to a herp conference, and we collected frogs. I got a nature paper out of it, so that's pretty resourceful. The point is that everywhere we found atrazine, we found hermaphroditic frogs and vice versa. So it's just a correlation, but we knew, for example, that we could go to Wyoming, raise this population in the lab, and they're not feminized, and put them in atrazine, and they are feminized. So we had the lab data to go along with the field data, and we published those data in Nature. We now have another study that we're just finishing up now where we've looked all over the United States. And what these dots are doing is these dots are following the North Platte, the Missouri, and the Mississippi River. So we have gradients of contamination, and we're looking for patterns of hermaphroditism, and hopefully we'll have that all published this year. Now, I travel a lot, and so I stay in a lot of hotels. And the only thing that makes me feel at home when I'm in a hotel is I can watch CSI. No matter what day of the week, no matter what time or where you're at in the world, CSI or some form of it is always on television. So I get my consistency there. Here's why I bring up CSI, because now what we want to do is, like CSI, we have the evidence that atrazine does this thing to frogs. Now, though, if we rope off our crime scene here, there's more than one suspect. Because if you go, and this is a shot taken out of a plane in Nebraska, if you look at a cornfield there, okay, and there's corn, and there's frogs growing, tadpoles, there's more than just atrazine on that plot. There's all these herbicides, these fungicides, these insecticides, and that's just on that one square plot. The next plot's using the exact different thing. The only thing they all have in common is that they all use atrazine. So we designed this experiment to ask, could these other chemicals be contributing? And you can see this is how it looks in my lab. Everything's color-coded. So all the students know to do is add the red, yellow, red solution to the red, yellow, red tank. It's blind. They don't know. I used to say it was double blind, but somebody pointed out to me that double blind would mean that the scientists and the tadpoles don't know what the treatments are. Although I guess technically that's true. Unless they've been looking at my notebook, the tadpoles don't know what treatment they're in as well. So I'm going to call it double blind. At any rate, we looked at these animals, and we looked at not just whether or not they were feminized or hermaphrodites. We looked at time to metamorphosis. And the interesting thing we found out was that when you start mixing these chemicals, it delays metamorphosis. In other words, one chemical doesn't retard their development, but when you start mixing chemicals together, you get these sort of additive effects, which could have consequences. What's more, as we showed, and I'm going to pop these data up and show you, is that we showed that when you start mixing the chemicals, you see a different relationship between growth and development. So in controls or unexposed animals, the older you are, the longer you take the metamorphose, the bigger you are. And that makes sense, right? The longer you carry your baby, the bigger the birth weight is. That's why you're not supposed to go over your due date. On the other hand, if you mix these chemicals, but not with any individual chemical, it's the opposite. They take longer to develop, and the longer they take, the smaller they are when they metamorphose. Now that's kind of an endocrine morphological observation, but it has consequences for these animals' survival. Frogs don't chew their food. So if they're smaller, then there's less likely to find food. And also if they're smaller, they're more likely to be food. And I know these look like great field shots, but they were set up in my kitchen and photographed. So there could be a real consequence on these pesticide mixtures being in the water. What's more, as we showed that of these animals exposed to the mixture in the laboratory, they were more likely to develop diseases like flavobacterial meningitis. And we showed that if you look at these animals exposed to the mixture, that their thymus has these holes in it. So their immune system's being broken down, if you will, when they're exposed to these mixtures. What we proposed and later showed was that we knew that stress hormones can also cause immunosuppression, decreased growth, retarded development, and inhibit metamorphosis. And so we proposed that what was happening with the mixtures is that the pesticides were causing a stress response. And the more individual pesticides you're exposed to, the more stressed you become. It's like if you have one bully picking on you, that's bad. You got two, you got three. The more you have, the worse the situation. They're each being added as an individual stressor. So to test that, we measured corticosterone, the stress hormone in frogs, and we showed that indeed these pesticide mixtures are increasing their stress hormone levels. And as a result, we proposed that's why you see these effects on growth and retardation of development and immune suppression. We also went back to some animals in the field and showed things like this. So this should be a liver This is an animal from an agricultural field. But if you look, these nodules in the liver, and this one looks like a smiley face wearing sunglasses. So that's why I always use that one. These are actually cestodes that have filled up this animal's liver. And this is kidneys from a different animal from the field. And they're kidney tubules, but the rest of these kidneys are trematode cysts that have filled up this animal's kidneys. But obviously have a real negative consequences on these animals. So we made a proposal in a paper that I published with my graduate students that there's several factors that could contribute to animals, frogs, not doing so well in agricultural areas. One, there are natural stressors that can increase stress hormones. High temperature, pond drying up, overcrowding. And these could be natural, or they could represent global climate change, drainage of wetlands, leading to the overcrowding, things that we've done. The desiccation or drying up interacts with the pesticides because as the water evaporates, the pesticides become more concentrated, and the stress hormones cause the release of fat-soluble pesticides as you burn the fat, which increases your internal stress hormone load. Those stress hormones then damage the immune system, like we showed in the laboratory, and lead to parasite infections like the kidney and liver damage, and also flavobacterial meningitis, which they could normally fight off. What we propose is that this interaction was incredibly important for a couple reasons. One is it means if you were only doing field work trying to figure out why populations of frogs were in danger, you would miss the interactions. All you would see were frogs that were too small, that didn't metamorphose in time, that had high disease rates. You wouldn't see the role that pesticides were playing without the laboratory experimental data. The other thing that I think made this model important was that the two organs most likely damaged by parasites are the kidney and liver. Now, why are those two organs important? What are the two most important organs in your body if you get poisoned? Like what do alcoholics die of? Liver damage or kidney damage? So by damaging the liver and kidney, you make it impossible to get rid of the pesticides, so the loads increase even more, which means you have even higher stress hormone levels, which means you have even more damage to the immune system, which means you have even more diseases and parasites that ruin the liver and kidney, which means you end up with even more higher parasite load. Well, that's all good, but how do you test that? Turns out that a natural experiment has been done, sort of natural, I guess, if you want to call it that. In California, the Salinas River runs south to north. Turns out that the Salinas River, the Salinas Valley, produces 85% of the country's lettuce. 85%, an incredible number. An incredible amount of pesticides are used on the Salinas River. What's more is most of the agriculture is on the north part. So you have a river that starts out clean. Santa Margarita is actually a watershed, and gets more and more contaminated. So we had this idea that we can go look at, and actually the red-legged frog lives on the Salinas River. It's a protected California frog. We can start in Santa Margarita, which is, no stressors. Water's, 1.5 feet deep. Very nice temperature. No pesticides yet. Further down in Atascadero, there's no pesticides yet, but the water's been drained off. That's a little pool that's about 2 inches deep, 98 degrees Fahrenheit. And what I'm showing you here is there's about 3,000 tadpoles in that little pool. So they're hot, they're drying out, and they're crowded. They're stressed, but no pesticides. Now further downriver, there's water again, 1.5 feet deep, relatively 25 degrees Celsius, but 100% of that water is agricultural runoff. So no environmental stressors, but now you're knee-deep in pesticides and fertilizers, so to speak. All right, so here's what that looks like on Google Earth. You have this pristine area in Santa Margarita, no stressors, no pesticides. You have in Atascadero, no pesticides, but extreme environmental stressors, high temperature, desiccation. And then further down, you have no environmental stressors, but the water is 100% runoff coming from the surrounding agricultural areas in Salinas County. So we did that experiment. We went out to these three areas. And first, we just collected tadpoles. And we asked, how are they developing? And it turns out that their development is retarded in Atascadero and Salinas, where there's high stress. Turns out their growth is retarded in both areas. And here's what that looks like. I'm a visual person, so. These tadpoles are the same age, same developmental stage, collected on the same day. And the only difference between this one and this one is that this one is growing in water that runs off of our food. No high temperatures, no crowding, no stress. This one is growing in water that runs off of our food, water that's tainted with pesticides. So then we tested, well, what's going on with their immune system? So we put out cages in situ, so out in the field, and then we did a real simple experiment. We just injected them with yeast. So we just injected them with bread yeast and asked, will they show a white blood cell count response? Will they show an immune response? It turns out in Santa Margarita, they're just fine with the yeast infection. Turned out in the Atascadero, at these doses, these are dead by the next morning. From all the high stress, now they have a disease. In Salinas, these are paralyzed, morbid, dead, morbid, dead. So all you have to do is be living downstream of water that runs off of our food, and you can't survive a yeast infection overnight. It says a lot of things. It also says that many diseases that we see that might be knocking off amphibians might not necessarily be because the disease is so bad, but when you're in stressful environments, you can't survive even simple parasites or simple diseases. So I want to remind you that the site that we looked at was Salinas, again surrounded by agriculture, because now I want to step away from the science a little bit, and talk about Steinbeck's East of Eden. That's my reference to A Tale of Toads and Men. East of Eden was written in 1952, and here's what Steinbeck wrote about Salinas and East of Eden. Salinas, he wrote, was surrounded and penetrated with swamps with tool-filled ponds, and every pond spawned thousands of frogs. With the evening, the air was so full of their song that it was kind of a roaring silence. It was a veil, a background, and his sudden disappearance, as after a clap of thunder, was a shocking thing. It is possible if in the night the frog sound should have stopped. See that dramatic pause? Everyone in Salinas would have awakened feeling that there was a great noise. In their millions, the frog song seemed to have a beat and a cadence, and perhaps it is the ear's function to do this, just as it is the eye's business to make stars twinkle. Here's what Salinas sounds like now. I haven't heard a single native frog call in Salinas since we've been working there. The tadpoles that we collect are from an invasive species, the bullfrog, to get washed downstream. And it's not a piece of scientific literature, but I think it's even better. Here's a literary artist that the frog sound and the frogs was such a part of the landscape that a literary artist wrote about the millions of frogs that were there 6 years before we introduced atrazine and these other pesticides, and now it's gone. It's not a piece of scientific literature, but it's evidence of how much we have changed a landscape that they were very much a part of. We published a paper where we looked at sort of several levels by which amphibian populations may be impacted. And I'm not going to tell you that atrazine is the ultimate cause, but environmental pollutants are intricately involved in pathogens, introduced diseases, atmospheric change, global climate change, habitat modification, and invasive species. They're a critical part, atrazine and pesticides, of the ultimate reasons for why amphibian populations around the world are in decline. And for those of you who don't know, as many as 70% of all amphibians are in decline. So I call this part of my talk From Silent Spring to Silent Night. Because in much the way that I think Rachel Carson's Silent Spring and the death of birds were a warning to us and the role that pesticides were playing in those bird declines, I feel that the fact that 70% of all amphibians, animals that have been around since the dinosaurs, are now disappearing, and that our pending, or almost in some cases here, Silent Night is also telling us something about ourselves as well. A colleague of mine wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. Now, you may not know it, but I've talked to you about more than one population of frogs, these experiments we've done. I've talked to you about multiple species, genera, they're not even in the same family. What I'm going to tell you now is that similar studies have been done in fish, reptiles and birds, and in mammals, so not just population, species, genera, family, orders, but every class of vertebrates that have been examined show effects like those that we've published in frogs. Now, to give you an idea of what some other points of view are, the Environmental Protection Agency says there is no direct information to assess what they're calling my hypothesis. However, here are all the studies that were done on amphibians at the time that I made this slide. There's even more now, on all these different types of effects. Here are all the studies that show no effects. Ironically, they were all produced by scientists that work for the chemical industry. What's more important is, if you look across vertebrates, here are all the studies in amphibians, reptiles, birds, and mammals that have shown adverse effects of atrazine. And these are all published. So somebody should get the EPA a library card, I guess. When you stack those up against the studies that show no effect, there's very few at all. So realizing this, a couple of years ago I said, somebody asked me if I had a book. I haven't written a book because I don't know who would want to read it. My mom would, but I don't know if anybody else would. That'd only be one sale. What I wrote instead is I said, I want to write a paper that actually looks at all these effects on all these different animals done in all these different labs around the world. And then I decided, rather than write that myself, I wrote to everybody in the world, and this is only a partial list, and I said, let's write a paper together. And part of the reason I did that is because this issue became very political. The industry started saying, well, there's nothing wrong with atrazine, there's just this crazy guy from Berkeley and his frogs. Now, I'm not going to deny being a crazy guy from Berkeley. I don't mean that my science is bad. Turns out that these guys just lost a court case. They settled out of court for $105 million. And the papers revealed from the manufacturer that they paid $10,000 to a psychoanalyst to determine if I was crazy or not, because I cussed at him on some email and some other stuff. Here's my first response. And I'm sorry, there's some young ones in the audience. First off, you ain't got to pay nobody $10,000 to find out if I'm crazy or not. You can ask me or anybody who know me, and they'll tell you that I'm crazy. Second is, there are only two possible outcomes when you pay $10,000 for something like that. One, they'll come back and say, no, he's not crazy. You just pissed off a black guy. Because, some people get those two things confused. The second possibility, because we're an emotional people. The second possibility is they'll come back and say, oh, yeah, he's crazy. Then you got to admit that you got your ass whipped by a crazy guy. So I'm not really sure what their strategy was, but I decided I wasn't going to play that game. We published one of the papers already from around the world. There's 22 of us from like 9 different countries or something. And here's what we found out. If you look at fish, testosterone is decreased in atrazine-exposed animals. That work was done in England. Here's my frogs, and now here's rats. Testosterone decreases no matter what animal you look at. In fact, we could show that testosterone reduction in response to atrazine causes the reduction in sperm. I've showed you that in frogs. So here's the testes from frogs that I showed you. No sperm in the testicular tubules. Here's a study from fish that was done in Belgium. Here's a study in reptiles that was study done in alligators down in South America. Here's a study that was done in both Africa and in Europe. Here's rats, sperm in their testes, feed them atrazine, no sperm. And I would have told you there was no evidence in birds, except a guy from Pakistan just sent me this. You feed quail atrazine, there's no sperm in their testes. So it's not just me, I still might be crazy, but it's not just me and my frogs. People from around the world are showing the same thing independent. I didn't even know about these studies when they came out. What's more is, my colleague Shauna Swan showed, here's control people, humans, and here's men in Columbia, Missouri, how much atrazine they have in their urine, men who can't get their wives pregnant because they have low sperm count. Now again, it's just a correlation, but look at this. Men who have atrazine in their urine have a low sperm count and can't get their wives pregnant, and no matter what animal we give atrazine, there's no sperm in their testes in the laboratory. What's more is, and I squashed the data down now, here's data from California from 1993 for field workers, and I'll squash it down now. Here's data from applicators. Men who apply atrazine in California have 2,400 parts per billion atrazine in their urine. These guys have 24,000 times the atrazine in their urine than we use to chemically castrate frogs that we know is associated with low sperm count in men in Columbia, Missouri. Imagine this. One of these guys could pee in a bucket. I could dilute it 24,000 times and use the atrazine in their urine out of their bodies to chemically castrate and feminize 24,000 buckets of 30 tadpoles each. We know nothing. Now this little boy who likes frogs is talking about environmental justice, because we know nothing about the health of these men, because they're 90% Mexican-American with life expectancies of 50 in many cases. On the other half of the equation, does atrazine turn on aromatase, which I asked you to remember, which induces estrogen, and that's going to cause vitellogenesis and oogenesis, or egg yolk and egg production, in amphibians, fish, and reptiles. And that's what we see. Here's amphibians, here's fish. This was done actually by the U.S. Geological Survey, and here's a study done by a guy in Canada showing reptiles. What's more is, just like I showed you, atrazine feminizes amphibians. The same thing's been shown in fish. You give atrazine in increasing doses, you see a decreasing number of males, i.e. they're turning into females. You see the same thing with amphibians. These data were produced in a laboratory in Japan, showed the same thing I've showed, and you see the same thing in reptiles in 2 different laboratories from around the world. So the feminizing effects are not just due to atrazine's effects in frogs, we see it across vertebrates. Now mammals have a sort of protection. They're not going to turn into females if you expose them to atrazine, but still I believe that my studies in this aquatic organism are teaching us something about this aquatic organism, because we grow up as a fetus surrounded by water just like my tadpoles do. In fact, estimates are now that we're exposed to 300 synthetic chemicals before we leave the womb, most of which we have no idea how they affect development and whether or not they're also endocrine disruptors. What you'd worry about in mammals, though, because aromatase and estrogen are involved, is mammary cancer and prostate cancer. It's already been shown that in one study with a p-value less than 0.0001, that people who live in areas that are contaminated with atrazine and drink that water are more likely to develop breast cancer than people who live in the same areas but don't drink their well water. What's more is, that's just a correlation, but if you look at rats, atrazine again causes a decrease in testosterone and a concomitant increase in estrogen, just like we've shown in fish, just like we've shown in frogs, just like we've shown in birds, just like we've shown now in mammals. And those rats have an increased incidence of breast cancer, of mammary cancer, which isn't surprising because aromatase and estrogen are associated with breast cancer. What's more is, because those are just rats now, if you take a human cell line, and this was published all the way back in 2001, if you take human cell lines that don't express aromatase or make estrogen, and give them atrazine, they start expressing aromatase and making estrogen. Just like we've shown in fish, just like we've shown in frogs, just like we've shown in reptiles, just like we've shown in birds, just like we've shown in rats. But now we're talking about human cell lines, human cells. I went to visit after I left a relationship down in Louisiana. Somebody was just talking about this place. This is down in a place called Cancer Alley. They have a pipe that runs right into the Mississippi. 1.2 million pounds of atrazine flow into the Mississippi every year. This has become an environmental justice issue for me for the following reasons. That community is 80% African American, has very high incidence of cancer. And it's not just Syngenta. There's tons of chemical manufacturers and oil refineries there. Here's why it's an environmental justice issue. These are the top 13 cancers you're going to get in the United States. In red now, 11 of the 13 are the ones that you're more likely to get if you're African American. What this shows now is the death rate or the mortality rate for Caucasian or white Americans in the United States. And this is corrected for socioeconomic and access to health care. What you're looking at now is the mortality rate for African Americans, and I can show you the same thing for Mexican Americans, from those same 13 cancers. Now, what does that mean? Does that mean there's a biological difference that leads to racial disparity? Or does that mean that if you're a minority, you're more likely to live in and more likely to work in areas that expose you to chemicals that we know are associated with these same diseases? It's hard to separate the two. In fact, you can't even start to do the studies. Great Komen for the cure, but put some money into prevention. You can't even get cell lines from minorities right now, cancer cell lines, to test the hypotheses. They're not even available, and those that are becoming available haven't been characterized at all. A word from my former sponsor. So, have we learned something from this aquatic organism about this one? One of the things we've learned, and my graduate student did this work, is that if you give a breast cancer cell atrazine, this is a gel showing that it's expressing the gene for aromatase, which means it's now going to make estrogen in a dose-dependent manner. Here's what I find interesting about that. The estrogen receptor is very important for breast cancer. In fact, you might ask yourself, right, because most women get breast cancer after menopause. You might ask yourself, why am I more likely to get an estrogen-dependent cancer after menopause when my estrogen levels at menopause are lower than they will ever be in your life? And it's because it turns out that these cancer cells express aromatase. And that aromatase makes estrogen locally that's not in your blood, but that estrogen causes those damaged cells to grow and spread. In fact, the local expression of aromatase is so important in breast cancer that the #1 treatment for breast cancer right now is a chemical called letrozole. Letrozole knocks out aromatase so that you can't make estrogen, and as a result, your damaged cells don't grow, don't turn into cancers, and don't spread. Now, how ironic is that, given that the #1 contaminant of drinking water does exactly the opposite? It turns on aromatase, it increases estrogen, it induces breast cancer in rats, and is associated with breast cancer in humans. Somebody thought about it. See, Novartis Oncology offers treatments for cancers that range from breast cancer. So this same company down here in Cancer Alley that produces atrazine also makes the chemical that blocks aromatase. So if you're living in the Midwest, taking letrozole to block your aromatase and treat your breast cancer, how is that impacted by this contaminant made by the same company that's in your drinking water that does exactly the opposite? I wrote a paper called The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. And they wrote this long, nasty letter to my dean. I'm just pointing out the facts. They make both chemicals. And on one website, they'll tell you Tyrone Hayes is crazy, aromatase has nothing to do with breast cancer. On another website, they'll tell you, we're going to treat your breast cancer by knocking out your aromatase, the same enzyme that we turn on. So, before I end, rats are used as a proxy for us, to measure things in the environment that might be bad for our own health. I work on frogs, but rats are, some people argue, more close to us and more important. Rats have been shown that it's been shown that when they're fed atrazine, they develop prostate and mammary cancer. They experience immune failure, just like we've shown in frogs. They suffer from neural damage when they're exposed in utero, just like we've shown in frogs. But more importantly, I think, something that changed my life, this little boy who likes frogs. If you expose rats to atrazine when they're pregnant, they will abort. The Environmental Protection Agency lab showed this and published it, not me. Of those rats that don't abort, another EPA laboratory showed that the sons are born with prostate disease when their mothers are exposed. Another EPA laboratory showed that the daughters are born with poor mammary or breast development, and that when those rats grow up, their offspring suffer from retarded growth and development because their mothers, who were exposed in utero, don't have proper mammary or breast development. Think about what I'm saying. This rat has retarded growth and development. This rat is affected by atrazine that its grandmother was exposed to. This rat never saw atrazine. This rat has retarded growth and development because its grandmother was exposed to atrazine. So when I think about my little girl, she's much bigger now, and the idea that my grandchildren could be affected by chemicals that we're applying today. Atrazine's been banned in France for over 10 years. The levels in the aquifer, the contamination, has not changed in much the same way that we're all still carrying DDT in our bodies and will for many more generations. When I think about that, this little boy who likes frogs realizes that I have a much, much, much bigger responsibility. This is a graph from a paper that just came out fairly recently showing birth defects increase when women become pregnant during peak atrazine contamination times. What's more is this paper also showed, and I'm going to read this to you, they concluded, not me, this is a group of people from the Center for Disease Control, that maternal exposure to surface water atrazine is associated with fetal gastroenteritis, particularly in spring conceptions. A former student of mine is a medical student now studying this issue, and recently it was reported that this rare, otherwise rare disease, there's a 9-block area in Britain where 9 babies in one neighborhood were born with this disease, with this deformity, with this birth defect. And there's high atrazine contamination in that area. So in many ways, and I was told I can't walk, which I normally do. I'm told I'm supposed to walk across the slide here and show you that I've crossed the line. But a little boy who likes science, a little boy who likes frogs turned scientist, has now also realized a much, much, much bigger responsibility. I've become active, actually, undergraduate, started this page on Facebook. Actually, I get a lot of my news about atrazine now from Facebook. I've helped with a number of petitions to ban atrazine. I helped Keith Ellison, who's now in U.S. Congress, write a bill to ban atrazine. He's just reintroduced that bill. So you can write to Congressman Ellison, you can write to President Obama, or write to Ellison, and that petition will go to Obama to help with that ban. And I just was featured in a movie along with Erin Brockovich to try and get much more of this data out to a larger portion of the public on not just atrazine, but pesticides and our loss of biodiversity in general. As you might imagine, my critics, many of which belong to Syngenta, but also other scientists, have called me some things now. Environmentalist is one. And I read the definition, because, I'm a Harvard-educated brother. I like to know what people are calling me. The definition said, any person who advocates or works to protect the air, water, animals, plants, and other natural sources from pollution or its effects. And I thought, who wouldn't want to be called this? This actually says more about the people who think that they're insulting me. Now, the manufacturer, though, wrote something different. They said in one of their press releases, Hayes, biologists, turn activists. I'm no longer a biologist or a scientist, I'm an activist. I turn, they said. And I thought, boy, this must really be an insult. So again, being an educated brother, I didn't jump to conclusions. I said, well, let me look up activists and see what I'm being called. So I went to the dictionary. And for activists, it says one that advocates or practices activism. And like many dictionary definitions, that wasn't very useful. So I looked up activism. You're not supposed to use the word in the definition. That's what my teachers taught me. But I looked up activism. It says a doctrine or practices that emphasizes vigorous action. And I thought, I don't mind being vigorous. What's the problem? But just in case, I looked up vigorous. And when you looked up vigorous, it says possessing vigor, full of physical or mental, exactly, full of physical or mental strength or active force. Still, I'm trying to find out what's supposed to be bad in there. So I looked up active force. And on the active, it says characterized by action rather than by contemplation or speculation. And I thought, that sounds an awful lot like a scientist to me. And I looked up action, and it says a strength or energy, especially of an exceptional degree, active power. And here I thought they were trying to insult me, and they were giving me a compliment. I'm indeed an activist. I'm full of vigor and active force. And now I've adopted a whole new philosophy. See, I was taught, let the science speak for itself. Don't get involved. Don't be an activist. And then I came across this quote. Those who have the privilege to know have the duty to act. And I figure I came from a family of $9,000 a year, but I gone to Harvard and Berkeley. I'm pretty privileged now. So I have a duty. Not only can you be both, not only do you not have to turn, if you have the privilege to know, you have the duty to act. And that's the philosophy that this little boy who loves frogs lives by now. And if you're wondering who said that, it was this guy. Thank you. Thank you very much. I realize I've taken all of your time and mine, but if there are questions, I'll be here. I don't know if we can answer some here, or I'll be outside. I'll be hanging around this afternoon, and we can talk, and any questions you might have, or criticisms, or suggestions, or anything you might have, I'm here for it, and I appreciate it. Thank you very much. Should we do some questions here, or should we all just go outside? I don't know what the rules are. Outside? Here, maybe I'll do like I do in my classroom. Some who want to stay here, raise your hand. Oh, we'll answer some questions here, and then I'll be available outside as well.
Incredible talk. Thank you.
Thank you.
I'm the executive director of the Xerces Society for Invertebrate Conservation, and we're advocating that the EPA take a stronger approach on all of these chemicals. You've talked atrazine here, but as you mentioned, all these other chemicals. This agency seems really, really stuck and really in a place where they're beholden to the chemical companies. I know the law that regulates pesticides was written with the chemical companies. Any ideas? get rid of them, start from scratch, or I know they do some other good things, but thank you.
Yeah, thank you. I've been very, as you might guess, very frustrated with my interactions with the Environmental Protection Agency. There are several problems inherent. Some of it is, I can say for myself, that I was sort of naive about what the Environmental Protection Agency really does. Some of the problems have been what people call the revolving door. There are a lot of people in industry who, for example, when I first started presenting about atrazine to the EPA, the scientific advisory panel was being run by one of the guys who works for the chemical industry, literally, who had worked with me on the atrazine thing. But some of the other issues are they don't really just regulate chemicals. They do what's called a weight of the evidence approach, and they balance, if I get rid of this chemical, what happens to the economy. So they're not just looking at environmental health, they're balancing that with other potential impacts on regulation. They also have to be in a position where they can't seem like they're going after one individual company or product, believe it or not. And the other thing that a lot of people don't realize is that the EPA hasn't done a lot of pesticide regulation at all. DDT, for example, was not banned by EPA. It was regulated, and individual states may have banned it, but it was never outright banned by the EPA the way people think that it was. I could go on and on and on for hours. I mean, the other thing is it's a political decision as well. So if somebody once put it to me, the president, of course, appoints the head of the EPA, and the first presidential caucus is in Iowa, where they grow a lot of corn and they make a lot of ethanol. That president's not going to go to Iowa on a platform of, I'm going to ban the #1 chemical used on the #1 crop in the U.S. I mean, so it's a much, much complicated, and at least when I got involved, had a very naive approach to what it is the actual EPA does. We could have a similar conversation about the FDA and other agencies as well. I wish I had some suggestions on what to do. I think that the bigger suggestions are, and one of the things I've been done is to hit these people from as many directions as you can. So I've been involved in consulting with the lawyers that just won a $105 million lawsuit, or they settled out of court with Syngenta. I've been involved in legislation. Tribunal 10, for example, that was banned by an act of Congress, not by the EPA. And I've been involved in working with the EPA. We just have to all be involved and put the pressure on.
Any studies out now or forthcoming on the reproductive success of the applicators? And on a related note, in this country, most of the applicators are male, whereas in other countries, they are female.
Well, the good thing about this country, I guess, is that we have, although they may be poor, better records than most other countries. I don't want to say there are no studies. There are no studies that I know of on the reproductive outcomes. Primarily, it's hard to get people signed up for these studies, when especially in the, when you have the pressure and the fear of being deported and all the other issues that come up with migrant workers, for example. It's very difficult to get those kinds of data and to get people to participate in those types of studies. So I don't know if it's been done. I'm not saying it hasn't, but I'm not aware of it if it has.
Green Diamond Timber Company uses a lot of atrazine around here in all our watersheds, including the one that supplies our drinking water for the Bay region. Is there any kind of way to have our water tested that would be reasonably accessible, without a lot of expense?
It depends on where you are. So I know that I actually get a thing from my water company. I don't know how frequently it comes, but it lists all the things that they test for. Atrazine is one of them. It tells you. So some places it's done.
They do it here. They do it once a year, I think.
That might be about when we.
And the sampling is probably unrelated to the application times.
Yeah,
I guess.
It would be, depends on where you are. Some places have labs that would do it as a service, as a public service, I've heard. But if you have to get it done by a private lab, it's quite expensive. A large part of our expenses was monitoring atrazine, because if you want your data trusted, you have to get a certified laboratory to do it. There are other labs that we can do it ourselves, but we want it to have our data competitive and not have it questioned as to how the analysis was done. I think it's like $1,000 a sample or something crazy like that.
Yeah.
There may be other ways to get it done. I know that I've been in states and places where they say, well, our county will do it for free if you ask. But that varies from county to county, or maybe municipal district to district.
Thank you.
Yeah, I was curious to know about the potential effects of atrazine compared to other chemicals, and why I've done a lot of research on that. Is it because it's one of the more widely used, or do you think it's more particularly has these dangerous effects, or is it more persistent in groundwater or surface water? What's the reason for that focus?
It's a good question. The first and most honest answer is because the company asked me to. I'd never heard of it. But the reason that it's been on everybody's radar is because, one, it's the number one, at the time was the number one pesticide in the world. I think it's number 2 now. It was and still is, the number one contaminant of drinking water, groundwater, surface water, you name it. So the one chemical I can guarantee you've all been exposed to is atrazine. And it's also persistence. It was believed to be not persistent, but as I said, in France, it's been banned for over 10 years, and it's still in the aquifers. Nobody's using it there for sure, and it hasn't changed. So it's high profile, again, because of its persistence, because of its ubiquity of contamination, and the amount that we use every year. And also now we know that it has lots of effects. The best thing I could say about atrazine is that we know it does a lot of stuff. For most of the 80,000 chemicals out there, nothing. If you look up pesticides, there's maybe 5 where you'll get a lot of developmental and reproduction, studies that have looked at the biology and how they impact the biology. The rest of them, nothing. At least not in the published literature. So there might be worse ones out there, we just don't know about them.
You mentioned at the end of your talk that there were time periods of increased atrazine contamination. And I was wondering if you could elaborate a little bit more on what changes seasonally or throughout the year as to why there would be increases.
Okay. In the Midwest, it's used on corn, and it's a pre-emergent. So before they plant anything, they use it to kill everything so that only the corn is coming up, so it's not competing with the corn. So the peak is when you get runoff off of those corn crops, and I believe it's around May or April. It's earlier as you go further south because they plant earlier. Here, it would be different, because here, it's not, here in this area, it's not because you're growing corn. When they cut all the trees down in the logging, then they apply it. And the application rate is very different. They apply a lot of it to a very small area, as opposed to in the Midwest where they're applying it every year all over, right? So it's a very different pattern of use in the Washington, Oregon, Northern California area.
Thank you. That is very true, especially because a lot of people get their drinking water from from forests here. But what I wanted to ask is, you talked about the European Union banning atrazine, and I think Israel also banned it. And I wondered if there are any studies about the decline in breast and prostate cancer in those regions, because that might be related.
I haven't seen, a few. So the European Union, a few other countries has banned it. Angola has banned it. Here's a country that has the longest standing civil war in all the world, and somehow they said, oh, we don't want atrazine, no. Don't ask me why. But I don't know that any of those studies have been done, and my impression is that breast cancer rates in Europe are significantly lower than what we see in the U.S. anyway. But I don't know that those studies have been done. And my feeling would be that you couldn't do them now because 10 years is not enough to actually see the impact, for a couple reasons. One is, I don't know the levels have changed in the aquifers, and 2, these kinds of effects aren't acute. So you drink something and you get ill. It has to do with long-term exposure. So my guess is you'd have to wait another couple generations before you start to see the cleanup, stopping using it now. And that's why I make the comment about my granddaughter's granddaughter potentially being impacted. Well, is there one more?
Yeah, I was wondering if you could repeat the name of the company that both produces atrazine and the chemical that reduces aromatase. Okay,
so let me clarify. So the original Novartis is the company I was talking about, and they were the merger between Ciba Geigy, the person who makes your contact lens cleaner stuff, Ciba Geigy, and a company called AstraZeneca. They became Novartis. So Novartis, for a period of time, made pharmaceuticals and they made pesticides. So Novartis, for a period of time, made atrazine and they made letrozole. In January of 2000, I used to have a slide that shows this. In January of 2000 was when they discovered that atrazine could cause breast cancer. In July of 2000, I think it is, was when they started selling this letrozole, so that's the period of overlap. And in November, they split up the companies, and one took atrazine and the other took letrozole. So just by coincidence, all in 2000, they discovered that atrazine causes breast cancer. They discovered an aromatase blocker for breast cancer, and then the two companies split. Your mic's not on, but I can hear you.
Is that, they're very
Yeah.
Do you know if there's any water filters on the market for at-home use that can remove any of these dangerous pesticides from your drinking water?
Okay, watch this. That's me not liking your question. Here's why I don't like your question. No, it's a perfectly good question. One is, I'm not paid a dime. If I had cut a deal with Brita Filters about 10 years ago, I'd be a rich man. Not that I'm worried, not that I'm concerned about being rich. If you look at the water filters you buy in the store, they even have printed on the side, removes the contaminant atrazine. Okay? But here's why I respond to the question that way, in all seriousness. One, it doesn't solve the environmental problem, right? So you can filter it out of your water, frogs, animals, everything else is still exposed. Two, it doesn't solve the problem for the people who are most likely affected. So those high levels that you see in factory workers and farm workers, those are all because of inhalation and absorption across the skin. So even if they had access to the information, and even if they had access to the finances to buy the filters, the people who are most affected aren't protected. The other reason is you shouldn't have to pay for somebody else's mess. And that's why Syngenta just settled out of court for $105 million with water companies to give them the money to actually have them clean up the water supply before it goes to the customer. So if the manufacturer is paying $105 million, which is nothing to them, think about the cost to us if we're doing it to clean up a mess that they're creating. So that's why. So I'm sorry to give you a hard time, but it's a perfectly good question.
Thank you.
Thank you.
Can you talk a little bit about your experience in the Scientists in the Field series, the book that you were featured in?
This is the Frog Scientist. It's a wonderful book. It's actually an award-winning book, the one that I showed here. Pam Turner, who wrote the book, did a wonderful job. She actually spent about 4 years with us, going into the field and into the laboratory. And the book not only the whole series sort of presents me as a person, and as I like to present myself in terms of how I got interested and why I got interested in what I do, but it also, in very clear terms, present the science that that a fifth grader can read and enjoy and understand, as well as an adult. So she did a wonderful, absolutely wonderful job with the book. I was very proud of the outcome.
It's a great book.
Thank you. Well, I will pass on the thank you to her. Yes. So the question is, what is about atrazine that makes it stay in the water supply? What happens with a lot of other chemicals is, one, they might either break down, and I'll give you my opinion in a minute, or they might bioaccumulate. So the fat-soluble chemicals like DDT, they end up in your body. They don't want to get back out. Atrazine's water-soluble. So the reality is, if I gave you a glass of atrazine right now, you'd drink it out, you'd pee it out by tomorrow. Right? So would a baby. The problem is, if you're a tadpole, you pee it out, but then you live in the water you pee in. The problem is, if you're a fetus, you pee it out, but then you live in the water that you pee in. The problem is, if you're a factory worker or a farm worker, you pee it out, but then you re-drink it again every day. So it's highly water soluble, so it's going out of the body. Here's the thing that I first started thinking about when I first got introduced to the idea of pesticides. You'll hear people say either, one, it has a very short half-life. And that's supposed to be a good thing, but that makes me think, wow, then that's a really potent molecule. Or you'll hear people say, well, this particular pesticide isn't very active. Then that tells me then it's going to be around for a long, long, long time. Because what we have to realize, somebody told me we shouldn't use the word pesticide, but biocide, is we're designing chemicals to kill things. And the talk for me, and again, I'm not saying we should get rid of all pesticides. We can have this conversation. But to me, talking about a safe pesticide is the equivalent of this term smart bomb. Right? You've designed something to kill things. Now somehow you're claiming it's smart and it's safe. What? Like something doesn't add up, right? And to drive through, and I used to see this when I was younger, and it didn't impress me the way it does now. To drive through Southern California, for example, and to drive past all these fields with the big red sign that says Pellagra warning, do not enter field. Poisons have been applied. And now somehow it shows up on your grocer's shelf and it's supposed to be safe. There's a big sign, and you see the trucks loading the stuff on the truck to drive it to the market. So somehow between that field, Pellagra, do not enter, danger, poison, and the few miles down the road, somehow now it's become safe. I think it's crazy. And I think we have to admit and realize if that's what we're going to do, in the same way that some medicines we take, right? We're poisoning something in our bodies that we want to get rid of. We have to admit that we're taking in poisons, and we have to be able to, like somebody was talking about GMO, we have to know what's going in our bodies, and we have to make a decision if we want it or not, and whether or not we'll take the risk. I don't like the idea of somebody else making that decision for us, because I don't think they make the right decisions.
probably not going to like this question, but anyhow.
Sure.
Your results really showed unsafe levels of a chemical, and I'm wondering if there's a level, any kind of level, that would be considered safe, or is it just one of these nanochemicals that is very bad for the environment?
So the question is there a safe level of atrazine? And that's I don't mind the question. That's a loaded question. For atrazine, I would say no, because we get effects of atrazine, and not just we, other labs, down at levels that are below what we can detect. Now, let me tell you this. Same thing's true for estrogen, a natural hormone. Very potent chemical. I'll also tell you this, which you may know. The safety limit that's set for lead doesn't mean that it's biologically safe. It means that's the level that we can measure. So even lead is harmful at levels that we can't detect. So we have to set the safety level at what we can measure. But our bodies can detect it at much, much lower levels than that. So atrazine is one of those. There may be other pesticides or other chemicals where that might be true, that if you're exposed below this level, and if that level were set, for example, to protect the unborn fetus, to protect children, to protect workers, to protect minorities, to protect the people who are more likely exposed, then I'd be fine. Or I'll give you an example that maybe I shouldn't bring up here. Or if that chemical has benefits that outweigh the risk, then I would feel very differently. Atrazine increases corn yield by maybe 1%. But that's $100 million. Atrazine increases corn yield by a small percentage, and we eat less than 2% of the corn that we grow. I would say that's not worth the risk. There are other chemicals, for example, that might be used, pesticides that might be used to control mosquitoes that carry malaria. I could be convinced for some of those, okay, spray it. If we're going to save this many children and this many people from malaria at the risk of some health consequences, then sometimes there's a balance, I think, that you have to reach. Atrazine is not one that I've been convinced there's a balance at, where you can use it safely and that the benefits outweigh the costs, in my opinion. Yes?
How do you test for atrazine in water?
There's a couple ways that you can test for it. There's something called an enzyme immunoassay that you can use, which we use in our laboratory. There are other chemical means for testing for it, like gas chromatography, mass spec, that only certain labs are certified to do, and that's the thing that costs a lot of money. Somebody showed me there's even like a home kit. Oh, this is relevant to the question I was asked earlier. Somebody told me there's even a kit that you can buy that's like a color changing kit or something like that. I'm not familiar with it, but somebody at a talk I gave once told me that there was. Yes?
Well, you talked about control frogs and frogs that were exposed to
atrazine. Mm-hmm.
Atrazine, yeah. How do you know that the control frogs were never exposed to atrazine?
That's a good question. So how do I know the control frogs are never exposed to atrazine? And it's related to your question. One is all the water gets sent out and tested to show that it's not control. The water that we use in our laboratory is, it's UV treated, reverse osmosis, double distilled, carbon filtered water, and then we put the salts back in it to raise the tadpoles, so it's pure water. That's one of the problems with some of the industry funded labs, is when they measured their water, their controls were contaminated with atrazine. So our water, like I said, is reverse osmosis, double distilled, UV treated, carbon filtered water with salts added back in. And we have it tested, and we test it in the laboratories. We do the experiments. And you're correct, because that range of doses that we use, if there's a splash from one tank to the next, you've ruined it. And you might have seen also in our pictures, we have the tanks all covered. And then the air is actually inside, so it's a completely enclosed environment. Same thing's true when we do our estrogen treatments, because that estrogen is active at levels that are similar to atrazine. You have to be very careful about contaminating. All of the glassware and things that we use is labeled, and they're only used for a certain chemical, so we don't wash and reuse glassware. Which is,
We had that pool party experiment where you had four control males for atrazine males, and you said that atrazine is water soluble. Do you think that the atrazine treated frogs were leaking atrazine into the pool?
Sure, I bet they were. But you're not going to get an impact in a few hours, right? Because the effects we're talking about are long-term developmental effects. And in fact, the effects on the tadpole, they have to be exposed throughout larval development, and then those are permanent effects. So even if you take them out of the atrazine, they don't recover and become normal animals. So it's a long-term, in much the same way that, for example, you won't get breast cancer if I expose you to this carcinogen once. It's long-term exposure to these chemicals that actually lead to diseases like breast cancer or some of the obesogens or prostate cancer and these things. It's a long-term, low-level exposure, not acute. I mean, don't get me wrong, there are chemicals I could expose you to and you would get affected right away, but those aren't the kind of effects that we're talking about in this case. Good question. Yes?
First of all, I want to thank you so much for everything you've done.
Thank you.
Second of all, I'm interested to know in what ways you can, you spoke to stepping off the iron pedestal, or the ivory pedestal, I'm sorry, and I'm interested in what mechanisms you propose to accomplish that.
One of the surprising things for me, again, I want to go with the little boy who likes frogs things. I never thought I would say things like, write your congressperson. I just never thought it would really matter. I never thought, but it makes a huge difference. I became very close with Keith Ellison and among other legislatures, and I learned they have formulas in their head that, oh, if one person writes me, that equals to 10,000 voters. I mean, they use these formulas to determine. And the other thing I learned is that if you work for industry, they have lobbyists. I never knew what that word really meant. They have people who sit in the office every day and are telling the politicians what they want to do. And if they and the wheel that turns is, if this company can pay this much money to have this person in my office, and that must be something that lots of people will want. So the more people who write to Congress people and say, I'm concerned about this, the more it gets on their head. The more it stays in their head that, oh, this is something I should do something about, because it's all about votes. I worked in Minnesota for years when they were trying to ban atrazine, and it's not that the legislators didn't understand the science. Keith Ellison talked about aromatase in his inauguration speech when he got elected to U.S. Congress. They understood the science. But what would happen is you would go to the, not the White House, what do you call it, where they hold the legislative hearings, and Syngenta would bring in 2 Greyhound buses full of farmers who would come in and just stand there. And so when those politicians voted on the bill, they'd look around and go, oh, I'm not voting for that. Had nothing to do with the science. Had nothing to do with understanding the science at all. It had to do with, if I vote for this, will these people vote for me next year? And so the more people who stand up, and even I didn't know, right? So these chemicals come up for review by the EPA all the time. If you go on their website, there's a place for public comment. They actually read those. They actually go, oh, yeah, how many people don't want atrazine to go back on the market? And even that has nothing to do with the science. They're looking for what people want them to do because they want to be back in office. So that, so I've become involved as much as I can in that way. I've become involved in the court case that was just settled for $105 million. I consulted. I didn't take a dime because I wanted to be able to say I did this because of my sense of social responsibility. I do everything I can to be, as a scientist, to be involved in public speaking, and there's a cost, right? So my dean will sit down with me and say, okay, you gave X number of talks this year, these don't really count. They count for 0. They could care less if I went to talk to Miss Brown's 5th grade class in Salinas. 0. So you have to make up your mind that this is something I'm doing for me, and I've had to have a real heart-to-heart about, because I had that dream, and I am I am the professor now. But I'm not the professor I thought I was going to be, where I'm worried about what they think of me and what my evaluations are about. That I'm looking at for my value is coming from somewhere else, because I've been told that. I mean, I've literally had my dean sit down and say, okay, yeah, you do all this teaching, you get good scores. We don't really care about that. This is at Berkeley. He just looked at me and said, yeah, I see you doing all this service, and you talked at the Presbyterian. We don't really care about that. They care about how many dollars you're bringing in, and that's it. So anyway, sorry, you got me started on I could go on and on and on. Well, if there's anything else, if not, I'll be around. Oh, one more. I think it was on. Now I think it's off.
Hello?
Yeah.
Oh, yeah. Okay, my question just is, do you think that there's a company out there that might be making another pesticide that the benefits would be better, less negative side to it than atrazine? So I've been involved in an effort, actually very peripherally involved because I'm not a chemist. There is a movement for something called green chemistry. And I actually just co-authored a paper that I actually had very little to do with, but I contributed, but wasn't a major contributor because, again, I'm not a chemist. And the paper is entitled "Designing
endocrine disruption out of chemistry. And so there's a group of chemists who are really working, who are using their knowledge of chemistry, which I don't understand, to design molecules that are not likely to interact in unwanted ways with biological systems. So there's a movement to do that. I wish I were more intelligent on the subject so I could explain to you how it works. And there certainly are, I would hope, companies out there that have a greater sense of responsibility. My naivete was once you work with a big company like this, if you found something bad, they go, oh, let's get that off the market. There are many, I can tell you from working with these guys too, there are many companies who already know that their chemical's going to be targeted. And they already have the replacement, but they just want to keep it on the market as long as they can as an economic strategy. And I think that was true of atrazine. I think when they hired me, they knew there was something wrong. The idea was you hired a scientist and you control when the data then comes out. Because there's no other place to get funding to do this kind of stuff except the chemical industry,
So
to be
it's
honest.
basically a money issue?
What?
Money? Is that like the...
I've become convinced that most things are, to be honest. Yeah, I mean, it's a money issue with the EPA, it's a money issue with the companies, and to some extent, it's a money issue with the politicians, right? Because if you do something, even imagine now, look at some of the debates that's going on now. Even if you do something that protects public health, if it had a big economic cost, do you think that person would be back in office? Probably not.
That's terrible.
So, well, and if you look at election campaigns, what is it all about? It's really, most things are about money, I'm convinced.
Okay, well, thank you.
So, sorry I couldn't be more optimistic. Well, thank you. I'll be around this afternoon, and thank you again for sharing this time with me.